Hello, everyone, and welcome to the inaugural ACS Medicinal Chemistry Letters Innovations webinar. I'm Lorraine Clark. I'm Managing Editor at ACS Publications, where I oversee 4 journals in the biological chemistry portfolio, including ACS Medicinal Chemistry Letters. We will begin the webinar today with a brief overview of the journal, the innovations article type, and introductions of the speaker and moderator. ACS Medicinal Chemistry Letters launched in 2010 with Dennis Leota from Emory University as the editor-in-chief. The journal offers a variety of manuscript types that are shown here in the slides, including innovations, and I'll talk about more on the next slide, and welcomes manuscripts that pertain to a broad range of subject matter related to medicinal chemistry, such as compound design and optimization, biological evaluation, drug discovery, drug delivery, imaging agents, as well as pharmacology of both small and large bioactive molecules. Now, turning to a quick background on the innovations manuscript type and the webinar. So we launched the innovations article type in 2016, and the aim of this manuscript type is to tell the backstory of drug discovery campaigns relating to the development of new therapeutic agents, and also to cover the discovery and development of new technologies that enable the identification of new therapeutic targets or advancements in acceleration of drug discovery programs. This webinar series aims to highlight research topics published in these innovations articles. And with that, I'm going to introduce our speaker for today, György Makara. He's the CEO of ChemPass in Hungary and has over 20 years of experience in the pharmaceutical industry, primarily in leadership levels in both the US at Neogenesis Pharmaceuticals in Boston, Merck & Company in Rahway, as well as in Hungary at AMRI, Cominex, and now at ChemPass. His expertise includes organic synthesis, medicinal chemistry, fragment-based drug discovery, drug design, and cheminformatics, and he has published over 30 papers in peer-reviewed journals and has also contributed to 10 patent applications. And now I would like to introduce our moderator for today, György Keserű. He is the unit head of medicinal chemistry at the Research Center for Natural Sciences and a full professor at the Budapest University of Technology and Economics. György is a member of the Hungarian Academy of Science and is a recipient of the Overton and Meyer Award from the European Federation of Medicinal Chemistry, or EFMC. He contributed to the discovery of the antipsychotic Vraylar that has been approved and marketed in 2016 in the US and the EU. Additionally, he developed virtual screening protocols for discovering both non-covalent and covalent ligands against therapeutically relevant drug targets and introduced library design strategies for fragment-based drug discovery. Before we begin, please note that we would love to get you, the audience, involved in the discussion of today's webinar topic. You can share your thoughts and questions in the Q&A panel, and we will address as many of them as we can after Greg's talk. And with that, I will turn it over to György to introduce the topic of today's webinar.
Welcome, and thank you very much for joining us today. In the next 2 slides, I just would like to summarize the huge impact of artificial intelligence technologies on medicinal chemistry. As we know that one of the key questions in virtually all of the medicinal chemistry programs is how to reach the target relevant chemical space effectively. There are many tools available, including automation or different synthesis technologies, synthetic platforms, even high throughput experimentation that would help us a lot. But in addition, or on top of that, there are AI methodologies that could contribute very significantly to reach the targeted chemical space. First, I would like to mention the de novo design, and this is basically the topic of today's webinar that might be useful for the hit identification, but very specifically in the hit to lead and the lead optimization. And the next slide, I would like to talk about on that. But before doing that, I would like to mention that there are many other places where AI can help medicinal chemists. One area is developing machine learning or deep learning models, just helping the selection or prioritization of compounds using different software packages or custom build or providers build methodologies. And another interesting field is retrosynthetic analysis that is not only useful for assessing the synthetic accessibility of compounds design, but also we can use these technologies for idea generation as well. In addition to the machine learning, deep learning methods, there are rule-based approaches, and today's topic, the ChemPass technology, belongs to the rule-based AI methods. So if we talking about the overall design for lead optimization, that is typically an idea generation task, then we have several methodologies we can apply. First, there is generative design using deep networks. There are many techniques available out there. And in addition to the fragment-based techniques, there are enumeration-based technologies. In addition to the simple enumeration that is available, for example, in the KNIME software suite, we have rule-based approaches, and SynSpace, today's topic, is a rule-based AI-assisted forward synthesis tools that would be very useful in the idea generation task. So here, just I would like to highlight the basic difference between different technologies mapping the pharmacological relevant space. Virtual screening maps this relevant space randomly. And as compared, de novo AI tools would help us to find the hotspots more effectively. And that makes the real DTMA cycle to a virtual one, and we can generate, we can evaluate, and we can rank different ideas very effectively. So now I would like to ask Greg Makara of ChemPass just to talk about the SynSpace technology.
Thank you, George. While I'm sharing my screen to be able to present the slides, I would like to thank the ACS first for selecting us for this inaugural talk of the Innovation webinar series. And hopefully many more exciting talks will follow ours in this series. So I hope everybody can see my slides now. So just a brief outline of the webinar today. I would like to start with a little bit of a discussion of our key technology in silico forward synthesis, which we apply for all kinds of design capabilities to solve lead optimization tasks. Specifically, I'd like to discuss in detail our generative de novo design tool, what we call derivatization design. And this is the technology and the case study which was published in ACS Medicinal Chemistry Letters this year. This part will be followed by several case studies, one which was published in the journal and a few other ones. And then hopefully we'll have enough time to show you a live demo of SynSpace and how we can accomplish the various tasks in a relatively quick and very simple manner. So brief introduction of our company. We are a platform service provider in preclinical space. The company was founded 5 years ago, and we have two different types of offering. One is a SynSpace design software, which is available for medicinal and computational chemists. This will be the subject of today's webinar, some of the capabilities. But we also have an end-to-end AI-assisted lead discovery platform, which is composed of the different design tools and several other tools we've built for an end-to-end solution to support lead optimization for our collaborators. There will be one slide describing some of the tools which are part of this platform. So the basic premise in what ChemPass is trying to facilitate is the lead optimization costs and timelines. All medicinal chemists are very familiar with the long timelines and the costly adventure to take a molecule from hit to a preclinical candidate stage. And I think even today with the modern tools, the industry and academia still works in DMTA cycles, where the cycle time and the number of cycles are critical factors to determine the overall cost and timeline of this process. In the DMTA cycle, synthesis is far the longest and most expensive component. A synthetic cycle can range between 2 weeks to even several months when there are some compounds which are very challenging in their synthesis. Now, in this process, there are several mindset problems which we are all familiar with, where it's very difficult to stop the effort. So number one is when we have compelling design ideas, which look very promising in models or on the computer, it's really hard to dismiss them due to perceived synthetic challenges. So in many cases, we know it's going to be difficult, but we still want to see the biological data. The second issue is once the synthesis of these compounds and other compounds have begun, it is extremely difficult to stop the process just because several months have passed, because there's always another route to make a compound. The number of potential synthetic routes are limitless, really, to any molecule. So these are difficult mindset issues which will impact these timelines. Now, in the last several years, we have really large virtual databases available which really assist lead finding very effectively. eMolecules or other providers have large commercial databases. We have Enumeration Real Database or SAVI from NIH, where there are virtual billions of compounds available for screening. But since chemical space is so huge, these have very little impact after lead finding, and we need really custom tools to start designing into the specific chemical space of our lead and hit compounds. So that's what we have designed SynSpace for custom ideation in synthetically feasible space. Current version includes over 300 virtual transformations, which are, I will briefly describe how this works. And I think it's very important in our effort that we'd like to create a software tool which serves as a bridge between medicinal and computational chemists, making preclinical research more effective and more efficient. And I think this collaboration is very, very important to move projects quickly. So in our software, there is no need for cheminformatics skills for chemists to be able to use it effectively, and there is no need for synthetic knowledge by computational chemists to do the same. So the design concept is in silico synthesis in the forward direction, which is, if you think about retrosynthesis, it's very similar, just we're going towards the forward direction and not going backwards. However, there are several challenges which arise from using this technology to the new direction. So the way this works in our SynSpace software, that we look at every molecule not as a product like retrosynthesis, but as a starting material. We'll look at where synthetic where synthesis can be done or selective chemistry can be done on any compound. Software will identify these routes, and the evaluation is done very carefully, exactly the same way as medicinal chemists will look at a building block, will look at functional tolerance, electrostatic, where it matters, regioselectivity, stereoselectivity, all these different things. The software will identify these reagents as potential reactants for reactions based on the synthesis know-how, which we built via AI-assisted rule-based technology. And then with that building block database or a reactant database, these reactants will be reacting virtually to create step 1 products. Step 1 products become naturally the starting materials for step 2. So we do the exact same assessment on all these compounds. Again, these will be reacting with a reagent set to generate step 2 products, and we can keep doing this in a forward direction. There are a number of different challenges arise when somebody tries to do this, and I think that's one of the reasons why we are not aware of a lot of other effort doing this in a multi-step way. There are several techniques like Savvy, for example, where one-step reactions are enumerated virtually, but we can really do this for many steps for different tasks. So one is obviously the rule-based AI technique, how to evaluate synthetic feasibility of reactants. So that's number one. Number two, how do you use this concept that you generate compounds virtually to solve actual lead optimization tasks? That's very important because you have to solve the specific tasks. And three, control of combinatorial explosion. That's very non-trivial, and we use very different solutions in our different software modules. So going back to the lead optimization case, this pictorial will show a very popular computer-aided lead optimization workflow when we generate a set of compounds, and we are going to start doing some filtering using more and more resource-intensive techniques. And at some point, somewhere around docking, depending on different docking tools, techniques, this can start becoming resource-intensive, like flexible docking is resource-intensive. And then we'll start paying a lot of price in terms of cloud costs and license costs to be able to generate a few molecules at the end with more and more precise, but more and more computationally expensive tools. And at the end, typically, we do retrosynthetic analysis today, and then some of these molecules will pass and some will fail. The issue here is that if we identify 35 compounds at the end of this funnel, and we have to throw out 15 or 20 of these due to a lack of retrosynthetic feasibility prediction, then we have wasted all these resources. These compounds actually represent half of our collection in this case, if it's half of the molecules. So we spent half of our cost completely useless because we throw out the end result. The other way of doing it is actually doing retrosynthetic analysis before we start the resource-intensive filtration. But doing full retrosynthetic analysis on millions of compounds or hundreds of thousands of compounds is not feasible, or it's going to also cost a lot of money. There are several scores like SA score, SC score, which, however, which are supposed to help, but they are not really useful because they are a measure of not synthetic feasibility, but really molecular complexity, and the two things are very, very different. There's a new score, RA score, which has just been published. This might actually be better, but recently I've just seen some presentation where there was noted that there's a lot of false positives even in this one, but that remains to be seen. So either way, we're going to have to spend a lot of computational costs somewhere to be able to validate molecules for synthetic. So that's why I call synthetic cost of de novo design. So why not just use a technology which will generate synthetically feasible compounds to begin with? In this way, we are not going to have the full retrosynthetic analysis issue here. We can do all our computational expensive techniques to filter down to as many compounds as we think is reasonable. And then we can do full retrosynthesis here. There may be one or two compounds which will be deprioritized, but really most of the molecules will actually do well. This looks like a much more advantageous workflow and certainly should be more cost effective, or there is little wasted resources in this. But really the question arises, how good would that chemical space be? Would it be at least as good as one we can generate with simple enumeration or deep generative design? So that's what we try to address with some of the examples today. So going back to the software, it has a lot of different modules to accomplish different tasks. Library design, we can do sidechain design. A lot of tools are available for scaffold design. This is really a key feature in our capability in our software. Retrosynthesis, library enumeration, multi-step, multi-reaction per step in a single enumeration can be done with our tool. Everything is going through the same synthetic feasibility assessment at every step, so this feasibility is assured. And we also have two different generative design solutions, and one of these is derivatization design, which will be mostly discussed today. But how would we use these different tools to accomplish lead optimization tasks very quickly and efficiently? So this is just a sort of a virtual case study where you have a hit compound and you'd like to get to some more advanced molecules. And there's a number of things one does during these sort of exercises in hit to lead and lead optimization, like retrosynthetic analysis, scaffold hopping, variations of building blocks, SAR development, and so forth. So we can do retrosynthesis, obviously, and so can a lot of other tools. But we can really very quickly do scaffold hopping with very simple tools, try to change the central scaffold or some of the other rings, do rigidifications, like turning a monocyclic ring to a bicyclic structure. And we can use either one of the scaffold design tools, which were on the previous slides, or we can actually use our generative tool, derivatization design, for this task as well. Very, very early, we can also try to do generative design to explore the chemical space early around our hit, because if we don't know too much, this is very valuable in SAR development. So we have these different tools to do this very quickly. Once we have like a more well-characterized and identified SAR, and we have scaffold structures, attention usually turns to optimizations of side chains, moving around heteroatoms, development of the lead series. And we have, again, a number of tools to do that very efficiently and quickly. One is library enumeration, where we can change the molecule at any position using the forward in silico enumeration tool. Again, we can do multi-step, multi-chemistry in every step. So this can be very, very efficiently done very, very quickly. We can also do just specific side chain variations. So once the scaffold is sort of taken care of, one can just modify the side chain specifically. And of course, we can do very comprehensive evaluation of the chemical space around this lead structure, which is now well characterized with our new derivatization design methodology. I think it's very, very important. I can't stress it enough how important it is that every compound is generated by SynSpace will always have all the relevant synthetic details, synthetic schemes, stepwise or in full, require reagent information, vendors for these reagents, predictive synthetic cost, and other features which are very valuable in ranking early. So just one quick example on a slide how the scaffold hopping tool, one of the scaffold hopping tools works, which we call one-click scaffold design just because of its simplicity. Really, the user only has to draw the current best lead or one of the lead structure, which has important feature they like, and then answer a couple of very simple questions regarding hydrogen bonding features, aromaticity, or ring size they want to change or not change based on the SAR at the time, whether they want to rigidify the structure further or not, and we must just press start. As a result, they'll get a scaffold list with properties. These will meet the 3D overlap criteria defined by the software, and also will have the synthetic schemes on a separate tab. One can review all or just part of the synthetic schemes for every scaffold with the ranking tools for building block availability, synthetic scheme ranking, number of steps, and so forth. That's simple. We also have the generative tools, and I think one of them, which will not be described, is a sort of a component-based generative design tool where we divide the compound to building blocks, and then SynSpace will attempt all the reactions in its arsenal to put together compounds which are similar to this. Again, every molecule, as a result, will have all the different properties and synthetic scheme data. And derivatization design, which is a much more comprehensive tool compared to the previous one, because this one only works for 3 synthetic steps. Derivatization design is limitless. You can run it as many steps you want. And with very simple setup, and again, it generates a very highly relevant generative space around your lead structure. So what is this? How does this work? This is described in our publication, which is noted up in the left top hand side. The concept is very, very simple. We divide any lead molecule or hit molecule based on the synthetic steps the user chooses for the molecule. We divide it into building blocks or reactants. So there will be, in this case, 4 reactants and 3 chemical steps, noted here as original sequences. So then, SynSpace takes over. It will look up what type of other reactions can these different functional groups do, which are present in these building blocks. By the way, actually, the user does not have to specify functional groups. You just put an R group here, the software will identify functional groups which are suitable for these reactions. But it will also look for other reactions these functional groups can do, at least one of the 2 partners. So we'll get a list of reactions for every step, including the original one and sort of related reactions. We can do the same with building blocks. We have the original building block, but for every reaction, we can generate products, how they would look like in a product when we look at all the reagent sources available for the design. We generate sort of product forms of these reagents, look at the similarities via multiple similarity measures, and we'll end up with a list of building blocks for every step and every reaction. So we have a multidimensional reaction building block matrix here, which will now SynSpace will use to put together final compounds based on the synthetic rules, of course, and the variation rules, which are defined by the user, how many variations you want to have on your final compound. The user will only have to specify the level of similarity and the number of variations. Everything else is done automatically, fully automated. And the synthesis and reagent vendor data, again, are fully included with every compound. So when we look at the case study in our publication, just taking one of the 8 ones which we published, there are 7-step syntheses here, which we uploaded into Derivatization design. And we specified, I'm sorry, I'm going to jump back here, that these 2 areas are more interesting, where the scaffold is present. So we added a lower similarity threshold versus a higher lower threshold at the outside 2 building blocks. And you can see the product distribution here. A high lower building block similarity threshold will allow very few reagents to be incorporated. Where we allow a larger range, there is the majority of the products come from. The product distribution will be really dependent on the number of building blocks available for the software to use. So the reagent pool, size of the reagent pool. The similarity range set by the user, you can see 0.7 lower threshold will generate much more analogs as opposed to 0.9. So very simple. Everybody understands similarity. And the reaction type. Obviously, a bimolecular reaction has a lot more opportunity to add more variations to a compound versus a monomolecular reaction, for example. Just to demonstrate what the product, 5,000 molecules or so generated by this very simple setting used for the design. We allow small variations here, so you can see sort of a building block swap. We allow more variations in the middle, so there are a lot more bigger changes. You can see that actually there are truncated molecules which will meet the criteria, but they are really relevant for our product types. You know, one could imagine this is a scaffold hopping, as well as scaffold variations here. And again, smaller changes outside where the similarity threshold was much higher. A simple change of the setup going to 0.7 lower setting everywhere, all 4 building blocks now have the same similarity opportunity, will change the product distribution as expected to a lot more even distribution. There are still differences, of course, because the reactant pool is not the same size, but you can see that we reach the desired variational sampling with a very simple change. So going back to our study, which we used to compare our tool to deep learning-based de novo design, we compared our capability to the 30,000 GON, which is a deep learning methodology, GON-generated library set, specifically designed for DDR1 kinase. And we looked at enrichment by the deep network versus derivatization design for this target. And the statistics are shown in this table, and there's more details, of course, in the publication. You can see that almost 1,400 compounds were used to train the deep network, which is a very significant number of compounds. We don't have that in many cases for most projects. We only ran 8 different derivatization design runs using different starting points from known crystal structures. Around the same number of compounds, 6,500 were selected for docking. And you can see the glide hit rate, 8% versus 34%, and the different glide docking scores, which are obviously less relevant because specific docking scores really don't track well with activity. But at the same time, I think for enrichment, docking is very valuable. We use this all the time for virtual screening. So there's a very significant difference in enrichment in favoring derivatization design versus a deep network. And this is not surprising. There will be more data followed later because deep networks require setups where user will allow a lot of changes to be incorporated. So by nature, a deep network will always generate a lot of molecules which are not very relevant to be able to create the diversity to make relevant changes for the project. Or there will be a lot of compounds generated which are very trivial for medicinal chemists. Very importantly, the number of new features discovered by the tools is 4 versus 6. So it's about the same. And synthetic feasibility is obviously very mixed for the deep generative tool. You can see some example structures from that set. Some are sort of all relevant smiles or valid smiles, but obviously not really relevant for DDR1. And some of them actually, which are more relevant, perhaps have long and difficult synthetic schemes. So very, very importantly, for the purpose of our tool, we have verified that we can rediscover retrospectively known prior art. Molecule 1 was generated by GON, was sort of the main finding of that particular paper in Nature. We can discover a molecule very similarly, it just includes one more solubilizing group with about the same glide score. And this is a completely separate paper where sort of a scaffold hopping was made from an ADMET liability to a more advantageous compound, and we can also discover the same feature in one cycle. We also looked at the chemical space generated by these compounds, by these technologies. And yellow is sort of the deep network design, and purple is our derivatization design tool. And you can see there's a main significant difference that there are molecules in the middle here which are really not sampled by derivatization design, but really sampled heavily by deep network. Moreover, when you look at the actual relative heat map of these compounds, the highest density of the structures are in this area. But when we plot the darker spots, which are sort of the docking hits on the top of all molecules, you can see that there are very, very few hits in this middle. So this highlights what I mentioned earlier, is that deep networks will always generate molecules which are completely relevant for the target to be able to create some interesting diversity, but for fewer compounds. So just looking at this one study, we would know that our sort of similarity-based generative tool has much higher hit rate for DDR1, have better docking scores, which I didn't talk about, but much lower docking scores for the top and for average. It discovers the same or more new motifs. It needs no large training set. You can do this when you have a single hit. Derivatization design is fully deterministic. As many times you run it, you always get the same result, which is not the case with deep learning methodologies often. It's very simple setup and control the outcome. We'll try to demonstrate that. And also we can do optimization, lead scaffold hopping, and so forth. And very importantly, when we control the synthesis at ChemPass and we do projects, we get well over 85% success rate for multi-step, 3 to 6-step chemistries in a very short synthesis time. And that's very important. So there's a very recent article on another DDR1 deep generative design study, and I think it's worth including. It looks like DDR1 is becoming a hot sort of virtual design target, where the authors used the publicly available reinvent technique shared by AstraZeneca very kindly, where the technology first generates valley smiles by a prior network, and then the authors used pharmacophore models-based scoring towards biasing the set to DDR1 type of inhibitors using an agent network via reinforcement learning. And at the end, of course, the user can get smiles of newly generated molecules. So the results were very interesting. The authors noted that molecules with pharmacophore score over 0.5, which is not very high, were much more abundant in the agent network, which shows that it really works well. It biases towards DDR1 versus the prior network, which is just, in terms of DDR1 relevance, is really random. When they looked at molecules which are sort of more relevant, 0.8 or higher pharmacophore score, they got 10,000 structures out of this 570. But overall, we would like to know that this is almost 8%, which is almost the same as we found in the other in our study for the hit rate. And I think it's very important that it shows again that there is a very high sort of false positive rate when one uses deep network-based generative design to create interesting new molecules. The authors selected over 4,000 compounds to visually inspect for synthetic feasibility, because that is not scored in the model. And 9 molecules were selected for synthesis, and 3 molecules had activity around 0.1 to 0.2 micromolar. And I think it shows those who are familiar with DDR1 inhibitor space that deep networks do a very, very good job at combining and mixing different functional groups which are known to work well. However, when one looks at prior DDR1 art, in this case, it's very, very quick to notice that there are very, very high similarity of these newly generated structures. So as a medicinal chemist, I think we would know that there is little structural novelty among these 3 hits. Of course, there could be more interesting structures in the other compounds which were not selected. So very quickly before the next slide, I would like to mention a few things which we have in our end-to-end discovery platform other than design. We've incorporated active learning tools to accelerate docking or other computationally expensive, more expensive techniques. So we only have to sort of process a small % of the actual molecules. We also have developed active learning for compound selection and ranking. And in our case studies, this tool actually sort of performs slightly better than medicinal chemists in selecting 3 different medicinal chemists were selected as a comparable study, and the technology actually generates more interesting compounds or selects more intelligent, slightly more intelligently from the better compounds than the more experienced medicinal chemists. We also have desirability scoring, of course, and deep learning and machine learning models. But I think what I wanted to really mention here, which is relevant for the next slide, is that we have developed a number of tools to analyze docking poses to eliminate false positives. I think this is very, very important. There were publications in the past by, for example, Vertex in this for kinases. And very, very recently, the Scheuket group published a paper on ligand strain energy analysis for docking. We've been using similar tools, but differently, and we evaluate a number of different factors: strain energies, presence or missing hydrogen bonds, binding poses, structural issues in ligands, and so forth. And this is one example here you can see, which obviously one can immediately notice that there is a distortion of aromatic planarity here for a molecule with a very, very high docking score. And this could be a source of a false positive right away. So we use these tools to automatically analyze poses and get rid of decoys, false positives. And I would like to know that well over 50% of the poses are typically flagged. So the reason why I showed this is on this slide that we selected one molecule, and I would like to know that the scaffold structures are slightly different from the actual ones because this work is in publication, going to publication. So but I think the concept shown on the slide is exactly well represented. So we selected one of the new motifs which we identified from our DDR1 publications in ACS Medicinal Chemistry Letters. And this was a very interesting compound because it has very high docking ligand efficiency, which we also compute, compared to ponatinib, which is a very well-known DDR1 inhibitor. But we noticed that the poses were flagged. So what we used, and derivatization design is also good for this, we used an exploitation run, generated 20,000 compounds, and we looked at the docking poses of these molecules, and we noted that pretty much all of these were also flagged. So this means that really this scaffold is sort of handicapped. There are certainly strains for all compounds, so this might not be a great candidate for synthesis, although despite of the excellent docking scores and docking ligand efficiency. So instead of pursuing this molecule, we went to one-click scaffold design to switch the scaffold. We generated 37 relevant scaffold analogs very, very quickly, and we identified several ones, but one slightly modified is shown on this slide as scaffold 2, which is comparable by docking score to a comparable representative of the previous scaffold 1. However, these types of molecules were not flagged. So again, we went back to derivatization design, did an exploitation run of 30,000 compounds. We actually used multiple chemical routes to the molecule. And we noticed that most of the docked compounds had no flags, or many of them had no flags. So this was a much better central motif to develop leads out of. Again, you can notice that just a simple cycle of generative design can really significantly lower the docking score, which may or may not track with potency, of course. And one wants to use FEP and other methods before synthesis in most cases. However, to sort of demonstrate the point a little bit, we selected one compound from each set, these ones and these ones, mid-range compound, not the best ones, somewhere in the middle of the docking score. One is flagged series, the other one is not flagged. And synthesis was done 2 to 3 weeks for the two compounds. And the flagged compound is completely inactive, and the other one is around 100 nanomolar, which is really a good starting point. Although, of course, one wants to notice that most likely there are much better compounds in our existing generative set as well. So this workflow is just maybe a demonstration on how quickly we can actually move a molecule through our different tools and with simplicity and get statistically feasible chemical space explorations done. Another case study, also from the very recent literature, an excellent publication appeared in ChemArchive this year on a generative deep network-based design study, where the authors noted that for a mature lead optimization project, where over 800 compounds were made, and 11 MP objectives were followed, the best compound met 9 of the 11 objectives. So that's actually not that bad, but they wanted to get better compounds. As we can deduce from the article, 11 QSAR models were used as a reward function, generative deep network-based compound generation, and there were 150 top compounds selected based on this workflow. 20 selected for synthesis, 11 synthesized and fully evaluated, and the best compound, and I think maybe there were 2 compounds which met all 11 objectives, which is a wonderful achievement for a sort of virtual workflow. And this is the structure of the best compound as identified in this publication. So we allowed our generative tool to investigate the same process, assuming, of course, that we have the same QSAR models, which for the POC study is irrelevant. And which we looked at whether our generative tool, derivatization design, can actually create these new features via virtual cycles. I think one thing is important to note that switching this building block on the right side to this one was most likely facilitated by the existing SAR, which is not disclosed in the paper. So we did not work on this part of the molecule. I think for medicinal chemists, for obvious reasons, this switch is sort of based on no SAR information. It's sort of difficult to imagine or sort of design. But we looked at the other part of the molecule, and I think as a summary, I would like to say that we generated 12,000 molecules in 3 cycles when we used the variation setting of 1, or 2 cycles when we used the variation setting 2, and we ended up with the exact same feature as one of the molecules included in this set. Of course, there are lots of other molecules which may or may not be also as relevant or more relevant or less relevant, but I think what's very important is that we can very, very quickly generate this type of molecules with derivatization design as well. In a single day, this can be accomplished with the tools. So how does derivatization design get to these compounds? This top scheme is a very reasonable route to this target molecule. It's a, I think, 5-step sequence. So when we submit the task to derivatization design, in cycle 1, when we use the total variations of 1, there are 2 relevant compounds, at least, which will be included, where the building block is switched, and when the chemistry is switched and the connectivity is switched from nitrogen to carbon. So these 2 compounds have these similarity levels as target compounds. When we use the same molecules as an input for cycle 2, we get this change of chemistry and connectivity in cycle 2 to generate this molecule with the level of, this feature in the final molecule with similarity setting of over 0.9. And the third cycle, when we use just one variation setting, will generate the final compounds, where the software will change the chemistry or the reductive chemistry to more elevated temperature and then be able to reduce the bicyclic ring as well. So as a summary, we can make all these different changes in the molecule with 3 simple virtual cycles. I think I'm going to skip this slide, although this is very important that software can generate virtual chemical space faster with fewer cycles compared to known publications. This example is on our website for those who are interested in this. What's very important, however, is that there is full flexibility with this technique at the user's hand. Some of these are very, very difficult to achieve with the deep networks quickly. The software works very well for synthetically difficult scaffolds as well as modular or linear compounds. If one looks at generative deep network design, most of those are actually done on more linear compounds. The method can handle those much better. The reagent source is highly flexible. The user can, with a change of simple reagent source, use in-house, commercial, off-the-shelf, at my CRO building block set, virtual building blocks, or customize the reagent set the user wants to upload. With a simple setting change, one can change the reagent source. And it's very, very relevant, because if I want to get a quick evaluation of space available at my CRO, which would be the fastest for synthesis, one wants to explore that separately. Third is full flexibility in what part of the molecule will be sort of evaluated. Of course, the full molecules can be changed with a simple run, or we can restrict the variations to separate parts of the molecules with a very simple setting. So this type of flexibility is very, very difficult to achieve with deep networks because the network will create all kinds of compounds, or it will require a significant change in the setup of the method to have this type of flexibility, which we need in lead optimization programs, I would add. And importantly, in lead optimizations, often we have key scaffolds in hand, maybe 2, 5, 6, 10 of these have been synthesized, and we want to explore variations just using these scaffold intermediates. This is very, very easily done using derivatization design, but I think it's much more difficult to achieve that sort of rigid sort of restricted evaluation of chemical space via deep networks. And lastly, how would we include this in a cycle? I think it's a very important question. So going back to the last example, we have our sort of starting point and this advanced lead structure. The user can apply the GUI, set up a sketch upload or query, built-in reagent sets, and get a derivatization design in 1 hour. Of course, the data can be exported into our models, evaluate the compounds, score them, and have a new candidate for design, one or several ones, and the cycle can continue. However, this can be fully automated using the SynSpace API, where, of course, the design is the same amount of time. However, the user does not have to draw or sketch or select, but since we have all the information from the previous cycle, a simple selection of the lead can actually issue the command to SynSpace via the API to run the next cycle. Very simply done. So I think I reached the point where I was hoping to show very, very briefly, of course, because we don't have a lot of time, how this actually works in practice. Scaffold hopping. We just want to draw our structure, mark the scaffold. If one hydrogen bonding is important, I'm marking it. Answer the simple questions. Want to change aromaticity or hydrogen bonding is important or not? Do I want to rigidify or do I want to vary the rings of the scaffold? And just press start. That's simple. You can choose whether you want to apply the Metcalfe filters or not. As I mentioned, the result page of such study will include the scaffolds and the properties. I will have to stress that we also have SureChemBL included in this. So we have hit count in SureChemBL, so there's a rough estimate of novelty included as well. If I wanted to run a derivatization design on one of these structures, I'll just set up the run. SynSpace will set up the entire scheme, as you can see, automatically divided the compound to steps and building blocks. It assigned total variation 1. I can change it if I wanted to. Here's my target structure. All I have to do is just press start. For the DDR1 case study, which we ran, these are sort of the fake scaffold. All the compounds are shown on a single tab. Every molecule, I can sort by similarity. I can look up building block list as well as synthetic scheme. I can go to e-molecules and check the latest availability data. I can refilter by properties, export, do clustering for hierarchical clustering or synthesis-based clustering, scaffold analysis. Going back to maybe the three-cycle study very, very quickly. Cycle 1 was set up with the original building block. Results are here. I just wanted to briefly show that highest similarity level, there's already a lot of variations. You can see we are using multiple similarity methods, which are useful. And very, very lastly, just wanted to show how easy it is to select reagents for library enumeration, perhaps. Nobody has to know about functional groups when they use SynSpace, because they can actually select reactions using drag and drop to a pool, and then use substructures or similarity-based other settings to select building blocks for different types of enumerations. So that's all I had time, unfortunately, for as a demo, but I think in summary, derivatization design is really a wonderful tool to do scaffold hopping, generative design with a high degree of flexibility, is deterministic, it can reduce the cycle count, and generate very relevant chemical space with high enrichment ratios. It's compatible with real or virtual cycles, it's fast, and it's ligand-based, obviously, TA agnostic, and I think our efforts are very important towards being able to allow medicinal chemists and computational chemists at the same time to effectively use these computational tools. So I want to just thank the ChemPass team for their devoted work to progressing these tools. Your attention, and my email address is here for those who have maybe follow-up questions or want to find out how they can access these capabilities. And thank you very much. Happy to answer questions.
Okay, so thank you very much, Greg, introducing us the SynSpace technology. And I would like to ask all the participants just submit their question using the Q&A window. Just before discussing the questions probably arriving, I have a question, Greg, about the maximal synthesis step you can consider during the derivatization design.
So derivatization design can handle limitless synthetic steps. It gets slightly slower to run with more and more steps, but not a very significant drop off. So we can do 9 to 12 steps, obviously, very close to about the same time. Timelines become much different when you use different variation levels and you allow multiple variations.
Okay, thank you. Now we have questions from the audience. And the first one is, the chemical know-how is always expanding with the new synthetic reactions being published every day. We know that. So the question is that, is the software able to accommodate these new reactions?
Yes, the synthetic transformation list is certainly expandable, and we updated it constantly. I think one important considerations we have is that when one creates virtual molecules with longer synthetic steps, it is somewhat important to rely on well-established chemistries, because more and more sort of dubious steps will make synthetic accessibility more and more questionable. So we think that a well-characterized several hundred size reactions pool is probably sufficient for a very thorough evaluation of chemical space using these tools.
Okay, we have a next question from the audience. Is the software capable of providing retrosynthesis?
Yes, absolutely. We have a retrosynthetic tool as well, and with the same one-click method, you can actually select a reaction sequence from the retrosynthesis reaction list and initiate your derivatization design with a single click. I would have to say just very, very quickly here, I hope my screen is still shared, that I can actually initiate a new design type, very different design types with a single click for multiple directions. So that allows quick moving forward in a virtual space using the different task solutions in the software.
Okay, another question from the audience. Do you stack rank difficulty of proposed synthetic routes? Chemists generally don't like lengthy synthetic routes for novel analogues unless they have high confidence in the biological activity. That's why it is important.
I fully agree, and we think it's very, very important that the shorter the sequence, the better. And very, very long sequences are more likely to fail. That's simple statistics, of course. In our software, there is a confidence score for every step. But which can be used for accessing, sort of evaluating the confidence level of a route. However, I would say that when we actually do projects ourselves, we'll always do a full retrosynthetic analysis on these few selected compounds for synthesis, because SynSpace is going to give you a lot of good routes. But I think when one starts spending money on synthesis, a good analysis is always in order.
Okay, thank you. So we have another interesting question, just starting from the ligand-based nature of the approach you have. So the question is that, do you think this software would be more useful for more established targets than for the newer ones?
Not at all. I think for any targets where you have some sort of idea what type of chemical space you want to generate, this can be used very, very effectively. The key is to be able to control the diversity of the molecules, and we have those capabilities. What type of tool you want to have afterwards to evaluate the generated molecules is really up to you. And if you have any information on the more difficult targets, you can apply those sort of philosophies or those models to limit the number of compounds and rank them.
Okay, so there is a relevant question about the possible deprioritization by pains or any structural alerts. So can you use these type of flags in the final compounds?
In the software, yes. In every user or as an organization as a whole, people can upload their filters, and this is sort of some of our filters. Software is fully compatible with smart patterns as well as simple structures. So this can be uploaded centrally or changed by each user separately from project to project. So these will actually remove irrelevant compounds. Now, in terms of pains filters, which are sort of flags rather than sort of unwanted moieties, we usually do that offline. To be honest, it would not be very, very difficult to include them in the software as well, but currently sort of pains is not part of it, although one can upload pains filters as metchem filters and remove those types of structures automatically.
Okay, another question about the patent activity. So where is the focus of the patent activity in this work?
Sorry, patent activity or
patent activity, yes.
Okay. So, if I understand the question correctly, in SynSpace, we built SureChemBL and commercial compound collection in there, so we can quickly evaluate sort of matching compounds in SureChemBL and in screening databases or in a corporate database. If that was the question, that's built into it. We don't have a full patent analysis feature in SynSpace sort of to guide the optimization. That would have to be done offline. But I think SureChemBL can certainly give a hint. If a lot of analogs around the scaffold return no hits, that might be a good sign. I'm not 100% sure whether I answered the actual question.
Okay, but do you consider the patent literature as well when generating the rules?
Oh, I'm sorry, I see. Yes, we do, but we are very, very selective with the literatures we use. While I think a lot of people are very, very confident about the patent literatures, honestly, we are not. I mean, we are very aware of patents, including molecules which have never been actually synthesized. Unfortunately, that's a known practice. So we are very careful in what type of literature we accept. But in our decision-making of the rules, we include relevant publications and relevant patents, and we do have our in-house rules for what we call relevant.
Okay, maybe we have a last question about what is the difference between the one-click scaffold hopping versus two-step general scaffold design?
Generative scaffold design will include schemes from commercially available reagents. So if there are similar reagents that can be applied in schemes original or new ones identified by the software, those will be incorporated. But one-click scaffold design is very focused on just changing that scaffold and trying to put together that molecule by any chemistry possible. So the two are compatible. There is overlap in the results, and potentially there are more scaffolds generated by one-click design as opposed to just using derivatization design. One more important difference. One-click design will not be able to go from a monocyclic scaffold to a spirocyclic scaffold, for example. It will be able to move from a monocyclic to a bicyclic, but not to a spirocyclic. Generative design will explore that as well. So there is an advantage to using both, and we actually typically apply both in a discovery project.
Okay, the truly last one is related to the known protein X-ray structure that is probably available for the target. So could you use protein X-ray structure from the virtual library via, for example, high-throughput docking approach?
Absolutely, we do that all the time. However, the generative and the scaffold hopping tool are both ligand-based, so it will require a ligand, which can be actually an artificial compound, if you will, a de novo molecule you generate by any tool, or you just draw it and looks reasonable. It doesn't have to be chemically or synthetically relevant as an input. That's very important, that the tools will be able to generate synthetically reasonable variations around irrelevant input data to make it synthetically tractable and relevant for lead optimization. So in summary, yes, you can, and we do this all the time, that the generated sets will enter docking and other tools as a sort of the sub-filtration methods.
Okay, so thank you very much. Unfortunately, we are a little bit behind the schedule, so therefore I would like to ask Lauren to conclude.
Yes. All right, well, that wraps up our program for today. Thank you, Greg and George, for joining us and speaking, and thank you, the audience, for attending and for submitting questions. Have a good day, everyone.
Thank you.